Hello class. I'd like to start today by reviewing what we've learned about cells. Here's the slide I showed on day 1 describing my approach for teaching this class. We are building up to the cell, starting with its most basic elements, atoms, small molecules, and macromolecules. Ultimately, these macromolecules will give rise to the traits of living things. So it's important we spend some time talking about what they're made of, how they're made, and what are some of their most basic functions. Okay, so on day 1, we established that all matter, including cells and macromolecules, were made of atoms. We went over the structure of the atom, and this helped us understand how atoms form bonds to make small molecules. Bonds formed with any given atom depend on the number of electrons, specifically the number of electrons in their valence shell, and atoms are most stable when their valence shell is full. These bonds depend on the relative electronegativity of atoms sharing the electrons in those bonds, where in nonpolar bonds, electrons are shared evenly, and in polar bonds, electrons are shared unevenly. We can also have bonds forming between ions that either give up or accept electrons, and they interact via this positive and negative charge. We also talked about water. We talked about why it's important for early life, its organismal function, but also because cells are mostly filled with water. Due to the polarity of water, which is a function of its shape and its ability to form polar covalent bonds, water molecules form hydrogen bonds with each other. Finally, on day 1, we started talking about carbon-containing molecules. These are important for early life because carbon was abundant, and it can form 4 covalent bonds with other atoms. Carbon will be the backbone of the 4 major macromolecules: proteins, nucleic acids, carbohydrates, and lipids. On day 2, we saw all the principles of day 1 in action and discussed how proteins were made of amino acids. We did activities to draw amino acids, and we saw what they look like after polymerization in a primary structure. Finally, we talked about how proteins will fold into increasingly complex structures. The next level of protein folding is spontaneous, and it depends on interaction of the R groups with each other as well as with water, and this results in a fully folded protein that can be functional. This week, we're going to take a look at the rules that determine whether or not reactions are spontaneous or not. We're going to be talking about the laws of thermodynamics and energy. We're then going to talk about how enzymes, which are a type of protein, speed up spontaneous chemical reactions. And in lab, you'll look at how pH and temperature affect this enzyme activity. And then finally, on Wednesday's class, we're going to move on to some new macromolecules, looking at nucleic acids and carbohydrates. So with no further ado, I'm going to move on to talking about energy. So really what we're talking about when we're talking about energy is the likelihood of a chemical reaction occurring. And that likelihood depends on 2 things. It depends on the potential energy of the molecules involved in the reaction and the amount of disorder in the system. So to talk about energy, we have to go back a little bit and talk about the laws of thermodynamics. So the first law of thermodynamics states that energy is never created or destroyed, it simply changes form. And an analogy of that can be a waterfall. So at the top of the waterfall, water or anything that's going to move down the waterfall has high potential energy. And as it moves down the waterfall, right, that energy is transformed, right, to kinetic energy. And then at the bottom of the waterfall is low potential energy, right? So again, at the top of the waterfall, we have high potential energy, and it moves down to a level of low potential energy, right, as it moves from something up high to something down low. And the energy isn't lost, instead it's transformed as kinetic energy as it moves down, and then once it hits the bottom, that kinetic energy is transformed again to heat or sound or some sort of mechanical energy. And the same kinds of energy transformations happen inside cells. So chemical bonds with high potential energies are those between carbon and carbon and hydrogen. And chemical bonds with low potential energy are those bonds between carbon and oxygen, right, polar bonds, and oxygen and hydrogen, again, polar bonds. And as molecules move from high potential energy to low potential energy, the energy lost it's not lost, it's transformed. Okay, so again, here's an example of the non-polar bonds that are weak bonds, but they have high potential energy because the electrons are shared evenly, and polar bonds in which the electrons spend more of their time orbiting oxygen than they do hydrogen, and those bonds are stronger and have less potential energy. But like I said, that energy, as it goes from a high potential energy to a low potential energy, isn't lost, it's transformed. And so in a test tube, this transformation results in heat, loss of heat, or in light, okay? So if you have a reaction in which you're breaking high potential energy bonds to make low potential energy bonds, that test tube will get hot. But inside a cell, and we'll talk about this in the next video, that energy isn't lost as heat, it's used to convert to create a molecule of ATP. The second law of thermodynamics is that entropy of a system always increases. And so here's an analogy for this. So this is my now 5-year-old when he was 2, and you can see he's emptying out our lazy Susan. And so a disorder of a system will always increase, right? And to create order again, you have to put energy back into the system, right? But this is the default, is increasing disorder. And then I had to use energy to put the Bisquick and the spaghetti back into the lazy Susan. Okay, so again, this happens inside cells. So we have here energetically favorable reactions in which entropy increases. So we have polymers, right? If it's proteins, we're talking about peptides, which are built of amino acids, or if we're talking about DNA, it's nucleic acids, or if we're talking about carbohydrates, right, it's monosaccharides building into polysaccharides. So anyways, we have polymers, and in the hydrolysis reaction, those polymers are breaking up into individual monomers. And a polymer going from a monomer increases disorder in a system. Energetically unfavorable then, when they require energy to put into the system, is joining those monomers together in a condensation reaction, right? So water comes out, but those monomers are joined together. And that's because the entropy of a system is actually decreasing, okay? So you have to put energy in. Now we have a mathematical formula that describes how these things of potential energy and how disorder of a system will influence whether a reaction is spontaneous. And we talk about that in terms of Gibbs free energy. And Gibbs free energy determines whether or not a reaction will be spontaneous or whether you have to put energy in. And so we calculate the delta G, or the change in free energy, during a reaction. And the standard free energy equation is delta G equals delta H minus T delta S. Okay, so what do those terms mean? Well, delta H is the change in enthalpy, or heat content, or potential energy of a molecule. And the delta S is the change in entropy, or the change in disorder. And T is the temperature in degrees Kelvin. Okay, so let's talk about what each of these terms means in detail. So let's start with enthalpy. So delta H is enthalpy. Enthalpy is the heat content of a molecule. Its value is based on the potential energy of a molecule. So if delta H is negative, then it releases energy. It's exothermic. If delta H is positive, the heat is taken up, or it's endothermic. Okay, so if delta H is negative, you can think of these molecules as favoring, they start off with high potential energy. And if delta H is positive, you can think of these starting off with low potential energy. For example, delta H of glucose, when we talked about carbohydrates, we talked about how glucose has a lot of potential energy. It has a very negative value for enthalpy. So it's negative 670 kcals. Carbon dioxide and water, right, which have less potential energy, like we talked about when we talked about carbohydrates, have positive values for their enthalpy. Okay, so this is just an example. Okay, so let's talk about the next thing in this equation. Actually, I'm going to skip ahead to entropy, right? Entropy is disorder of a system. A positive delta S means entropy increases. And again, this happens when we're breaking things apart, when we're creating disorder. And a negative delta S, meaning you've got to put energy in, means that entropy decreases. So again, taking monomers in a condensation reaction to build a polymer. And this all depends on temperature. So whether or not a reaction will proceed because of disorder matters what temperature the reaction occurs at. So here's again our free energy equation. So let's say we have our delta H and our temperature and our delta S. What is the value of delta G mean? Okay, so delta G, just as a reminder, is free energy. And if the value you calculate is negative, okay, it means that the reaction is spontaneous and exergonic. Okay, so it's going to occur if this value is negative. So that, hint, hint, that might be on the quiz. If the delta G is positive, then the reaction needs energy to occur, and it's endergonic. Okay, and an easy way to remember this, that I remember this, is exergonic. X is the energy out. So that means that if it's an exergonic reaction, it's going to be spontaneous. You don't need energy. And endergonic, for energy, means that you need to put energy into the system to make the reaction occur. So here's what this would look like on a graph. So we have free energy on the y-axis, and here you're starting with a molecule that has high potential energy, and your products have low potential energy. And this means that the change in delta G is negative. Okay, so those reactions are exergonic. You X out the need for energy, so this reaction will proceed spontaneously. If your molecules have low potential energy and you want to create a molecule with higher potential energy, then you have to put energy into the system, right? You have to, this value, this delta G is positive, and those reactions need energy. They're endergonic. We can think of this as another way by just applying some values. Okay, so this is all conceptual. Let's try it with some math. So if negative, the reaction will proceed spontaneously and be exergonic. Okay, that's because a negative, right, minus a positive is usually a negative. So if you have a negative value for delta H, it means that the molecules that you start with have higher potential energy, and that if you have a positive S, that means that disorder increases. Okay, so more potential energy and more disorder. So, for example, right, negative 10, a negative value, minus 20 is negative 30. Okay, so another instance is if positive, if the delta G is positive, then the reaction needs energy to occur and is endergonic. That's just a reminder. So and that's because a positive minus a negative is usually a positive, okay? So this means that a positive value would mean low potential energy for the enthalpy and less disorder, right? Because this would be then a negative, okay? And let's do this math. So a positive value of 10 minus negative 20, right? Double negatives equals a positive. Then this number ends up being positive. So this reaction is going to need an input of energy. If only one side of this equation is unfavorable, you have to do the math, okay? So if the delta H is positive, meaning that it's endothermic, you have to put energy in, but the disorder increases, it is more likely to be favorable at high temperatures. Okay, for example, okay, the delta H is positive, so less potential energy, minus, let's say this reaction is occurring at 1 degree Kelvin, and the entropy has a value of 5. That means that this value at the temperature of 1 degree Kelvin, this delta G is positive, so you have to input energy in, it's endergonic. But if you increase this temperature to 5 degrees Kelvin, then the reaction ends up, or the math ends up being 10 minus 25, which equals negative 15, and now our delta G is negative. If it has a high heat content, right, so that delta H is negative or the reaction is exothermic, but the disorder decreases, then it is more likely to be favorable at low temperatures. So this is just another example. here we have this reaction occurring at 1 degree Kelvin, so -10, we have a negative value, right, because it's exothermic for the enthalpy, and then minus -5 equals -5, so it's this reaction, it has a negative delta G, it'll proceed. But if you have the temperature increased, right, so at to 5 degrees Kelvin, right, so -10 minus 25 equals -25 equals 15. So now the reaction is endergonic. Okay, so in this case, it's minimized at low temperatures. And I'm not going to necessarily ask you to predict, right, which one which one temperature matters. But what I want you to know is that reactions in which the molecules have high potential energy and disorder increases are more likely to be favorable. And reactions that have start with molecules that have low potential energy and disorder decreases, those are less favorable. So for most reactions to proceed, you have to break one or more chemical bonds and others have to form. And so substances have to collide in a specific orientation to allow those electrons to interact with each other and break bonds and reform others. So if you have two molecules you want to interact with each other, right, they may or may not interact if they're far apart in a big solution. But the more of those molecules you have, and the higher temperature, which is going to increase the number of collisions, the more likely you are to have a reaction to occur. So that increases your reaction rate. So let's, now that we know that temperature and concentration also matter, let's just revisit this delta G with a few calculations. So here I have the hydrolysis of ATP, right? So we're going to add water to ATP and you're going to end up with ADP and inorganic phosphate. Okay, so I have the values for our Gibbs free energy equation. So delta G equals delta H minus temperature and multiplied by delta S. So temperature, I didn't mention this, is in degrees Kelvin. And so you can convert Celsius to degrees Kelvin by adding 273. And then this reaction occurs at 20 degrees Celsius. And the change in heat, or the change in enthalpy, is 19,070 cals. And the change in entropy, or delta H, is 90 calories per K. Okay? So pause this video and do the math. Okay. So here we go. So we'll start with plugging in our values. So we have here our delta H, which we'll plug in here, and then we'll subtract that from the temperature, which is 20 plus 273, multiplied by the delta S. Okay, so here I just combined the temperature to be 293 Kelvin, and then our Kelvin canceled out. Okay, so we're left with, and we can multiply these together then, and so we're left with calories and calories on this side. So you just do the math, and your delta G ends up being negative 7,300 cals per mole, or, and this should be kcals per mole, negative 7.3 kcals per mole. So because this is negative, this is a exergonic reaction. This reaction will proceed spontaneously. So nonpolar bonds store potential energy. That's one of the things that I really want to point out. And increasing a disorder, i.e. dispersing molecules, breaking apart polymers, is energetically favorable. And so in the next video, we're just going to talk about what to do with that extra energy, right? When, let's say we have these exergonic reactions, right? We don't just release that energy in a cell, we couple it to other things to make things happen.